They evaluate you. They use someone who is doing the same thing you are doing. So mentors and preceptors are very different. Again, I never assign mentors, and mentors should never be assigned. They are chosen between both you, the trainee, and the mentor, or the mentee and the mentor. And this was one of my very first ones who, and again, along the lines of what a mentor is, he actually is Chinese. He was a geo-oncologist who started at my residency program as a new attending. He's Chinese, but he's actually born and raised in Brazil. So he's Brazilian. So we had that Portuguese connection. So again, although he was someone that I aspired to be, and which fortunately I become, we had a lot of other things that connected us, and his goal was to make it better. And so for in the US, to be a geo-oncologist, you have to do a fellowship, which is extremely difficult. There's only 50 spots in the country to be accepted to be geo-oncology fellowship program. So he was very helpful and instrumental in guiding me and helping me get to where I am now. Then the first thing, this was also during residency when I first dipped into research. I've always been someone who questioned things and wanted to find answers and know why things are why they are. And my first dip into research act taught me a lot. It was a randomized trial. There was at that time, for those of you who never went through OBGYN, in order to induce labor, there was a process of people were putting Foley balloons into the cervix to try to ripen the cervix. So really there was no randomized data. It was all just a single institution experience. So for the first time ever at my residency, I was the first one ever to do this. I identified a clinically relevant question. Does this balloon that we're doing actually better or faster than what the standard was use a prostaglandin gel? So again, this starts the message that when you're doing research, you want to identify, for me at least, a clinically relevant question. Is this question that I'm going to try to answer, is this going to help me learn more about the patients I care for? And will it help me care for my patients better? And that's guided my research decisions all along the way through all those papers that you mentioned that I've published. So the first thing when you do research is you need to know the background. You need to know the history. To just think that you just came up with the greatest idea in the world and start doing research on it may be a mistake, because it may have been researched 10 years before and found to be not so helpful. So you do an extensive background review of the literature. Once you do that, you have that forever, that background. So like writing chapters and review articles is as painful as it is, and I don't do those anymore. I usually have the fellows do them. It is a great way to get the background knowledge that you then know everything that's kind of been done in that area. So then you can see where the next questions need to be. So what we also then is you learn that for a randomized trial, you have to have some basic assumptions, which come from your background review of the literature. So the control arm we had was labor induction time and prostaglandin arm. The experimental was did the Foley make it better? And then you have to come up with power, confidence, and one-to-one randomization. Proper statistics. So I learned about the need for this, consent process, all things that took a very long time. This is months in the process. And then speaking of the months is the painful IRB process, which I still go through every day. Fortunately, I have the fellows usually be the point people for that, so they can learn, but also takes away some of my pain. And then, we have to run the randomized trial and ensure proper randomization. So what I learned from this trial is all of these things for the very first time in my life. And then what I really learned is how even though this was a randomized trial, how messed up it can be. So just calling something a randomized trial doesn't mean that it's going to be done well. It's a very important concept when you do randomization or randomized trials, is that you don't want the investigators to know what sequence is coming up. Meaning, if you have treatment A and treatment B, you don't want the doctor to know, or the patient to know, what if you're going to randomize that the next one's going to be treatment A, because that might bias them to say, well, I don't really want them to get that treatment arm, so I'm not going to put this patient on there, or I'll wait to the next one. That's a huge flaw. And when you do a randomized trial, you have to describe how you concealed the sequence of randomization. There's many ways to do that. A basic way is to put the sequence you generated. You have, let's say it's a 100-person trial. You put it 100 envelopes lettered that you can't see through, because they can see through it. There's been stories of people looking at the light to see what the sequence was or the randomization arm was. And this does happen, because this happened with my trial. One morning I came in and the envelopes are in order. So you have to take one, you have to open it, you have to put the patient's name on it. It transfers a carbon copy to the inside. I came in and there were 2 envelopes opened and put back, and the 3rd envelope was taken. So whoever the surgeon, the obstetrician was, who was randomizing their patient, opened the first 2 envelopes, did not like what arm that was, put them back, and then took the one, and like that, it was whatever arm that they thought they wanted to give this patient. Completely ruined the trial. I actually stopped it, never moved forward with it, never published it. That completely ruins the trial. That's not random allocation. That's a highly selection bias by the surgeon. So from this moment on, created my so critical view of everything that's published out there. But in order to be critical and understand what's published, you have to understand statistics and trial design. Otherwise, you just cannot be critical. If all you do is read the results and then the conclusions, and don't even know anything about the methodology, you don't know what you're doing, is my opinion. Learning statistics is as important as learning how to operate if you're going to be an academic GYN oncologist or an academic surgeon. There's just no way around it. Otherwise, you're just trusting that things are done correctly. But this is what started, for those who know me, my critical view of publications in the literature. So another thing that's important is, always in the line of always doing what you think is best for you and that you like is, I went to OBGYN and obstetrics and gynecology thinking that's all I would do, but I didn't like it. I didn't think it wasn't for me. So I was thinking of leaving to go to surgery. But that's when Dr. Lou, my mentor that I had mentioned, we were talking, and I told him what the concerns were. And he said, why don't you look into oncology, which is extremely difficult. I wasn't at a very competitive program, but he helped me. And I was going to make a big change and go to a whole nother residency. And this is also something that you need to decide. If something isn't working for your life, you need to make a change. And making a big life change is always scary, but what's even scarier is regret. You don't want to be 10 years later in your career and say that you regret what you've done. I don't regret any of my career, as difficult as it is, as challenging as it is. I don't regret anything I do. If you ever regret what you do, then it's time to stop and do something different. So I did, after my residency, I did my fellowship, which is required in the United States to be a gynecologic oncologist, a 4-year program at Memorial Sloan Kettering. I did a resident rotation first, which helped me get the fellowship a little bit. And then my fellowship in GYN oncology was from 2000, 2004. This is when I met another great mentor, Dr. Barakat, also happens to be son of immigrants. My mother was German, father was Lebanese. And his is a great quote that he's always said, and I also take this to heart. If you surround yourself with the best people, support them with whatever they need, you will succeed also. So my success now is also a lot by how the fellows are succeeding in my junior partners. And how others are succeeding that I may have the opportunity to interact with. So this has also been a guiding principle for me. So work with successful people always, and people that want to move forward, and stay away from folks who are all just about themselves. This is also my very first publication. This is 19 years ago. Well, this is my very first publication where I was the first author. This is under another mentor of mine, Dr. Jeff Boyd, is a GYN oncology fellow. We spent 2 years in the molecular in our lab doing. My focus was on P53 mutations back then. That's how old that research was, looking at P53 mutations in early stage ovarian cancers. But this is my first publication. But what I learned from this, and you always need to learn every step of the way from everything you do, is that this was a very relatively small paper looking at preoperative C125 levels and those who had hereditary versus sporadic ovarian carcinoma. And we asked our statisticians to help us with the stats. They started coming back and giving us a lot of trouble, saying this is too small, you can't do this, all that theory. And we just realized that we understand all that, but just give us the statistics, give us a p-value, please, so we can this is all hypothesis generating. That process was so painful to me that I decided from that point forward, I'm going to learn how to do statistics on my own. I self-learned. I worked with the PhDs that were under the main statistician, because they were younger, about my age. I started calling them, asking them, help me, take me through this. And I took some courses, and I basically learned my own statistics. And now I teach all the fellows. Learning your own statistics is critical to understand that the statistics that are being done by others is correct. And it makes publishing so much easier. I don't have to wait for a statistician back and forth to get my data analyzed. I can do the majority of my data. Now, for larger scale kind of analyses and certain types of statistical techniques, I don't do, but if this is for our standard stuff that we do, at least clinically. So learn statistics. Even if you don't do it, you have to know how to do it and what it's all about to make sure you are doing it correctly and that you interpret others' statistics correctly. And reviewing thousands of papers now from multiple journals, I can tell you that first thing I focus on is not the introduction, because I don't care about it, because I'm a GYN oncologist. I know what the background is for all these papers. I focus on the methods first. If the methodology is not correct, I basically will reject that paper. I don't even read any further. That's how I can read a lot of papers too, because I will just toss a lot of papers out when I start with the methods. If it's crap, I mean, if it's really not good, I don't even read any further. Then I look at the tables and the figures. If I can get the message from looking at the tables and the figures, that's a good paper. If I have to find out too much description in the text, that's not as good of a paper. This actually was my experience in the lab. This was published in PLOS One many years ago with myself and two co-fellows. We did this. We all are listed as co-primary authors. And again, along the lines, even if you're doing molecular oncology, I think you need to really try to focus on questions that will have some translational, which at the time, translational research was a big sort of catchword. Everybody wanted to do translational research. But what this really is, if you're doing this in oncology, especially, really try to focus on things that can have true impacts moving forward in learning about cancers, no matter what they may be. And we're very proud of this. This was before the tubal hypothesis, looking at the natural history of epithelial ovarian carcinoma. We microdissected out the single cells from the inclusion cysts, from the surface of ovaries, from prophylactic BSO specimens. We looked at genetic changes, and we were able to find some early genetic changes within the cystic inclusions of the ovaries that had high risk for ovarian cancer, meaning they were prophylactic specimens. So very proud of that. Now, I don't do molecular research anymore. I'm mostly a clinician and clinical research. So after my fellowship, I went to New Jersey Medical School to start a career as a faculty member. I became an assistant professor. This is in Newark, New Jersey, where I was born. So I had some ties there. I went there and I stayed there for about 3 years. I was the director, actually, of gynecology for a year before I came back to Sloan Kettering. But what I learned here also, especially if you're doing research, is you can only use the resources you have at hand. You can't make things up. So where I went in Newark, there really wasn't a great GU oncology program. So they didn't have a lot of good cases, a lot of background, but they did have some resources that were pertinent to the area. It was an inner city hospital. So at that time, HIV and cervical cancer was a very big topic, how they interrelated. So this was the first publication as faculty that I did. And this is the only paper that ever came out of that institution at the time for GU oncology. We took care of a lot of patients with HIV, and we took care of a lot of patients with cervical cancer. I'm sure it's very similar to Brazil at the time and even now. And so, what I was realizing is that those who had cervical cancer just seemed to be more immunodeficient. So it wasn't just having the virus that mattered. When this paper was published in Cancer, actually with an editorial, I was a young faculty, so I was very excited about that. And we basically found was that, those who had cervical cancer and who had HIV had lower CD4 and lower viral loads. So if you then categorize patients as immunocompetent versus not, those with cancer were much less immunocompetent, meaning they either had a low CD4 or low viral or high viral load or both. And if you had a normal CD4 count and a low viral load, you were less likely to have cervical cancer. So, something that was important and at the time, and it was published in Cancer with an editorial by a well-known senior person at the time. So I think this was a helpful paper, and it's a question that I had that was clinically relevant. I was there for 3 years, and fortunately, Dr. Barakat asked me to come back to join the faculty at Sloan Kettering, and that's where I've been ever since. I came back in 2007, specifically with the request to build a robotic program. I also was asked to be the principal investigator for the GOG, which is a national cooperative group where I did both of those. I've built a robotic program. I became an associate professor and member in 2010. I took over the fellowship program in 2014, the director of all MIS for surgery in 2014, and I'm a full professor as of 2015. Now, MSKCC is not a university. It's a freestanding cancer hospital. So they don't give out professor titles. They give out member. It's called member, assistant, associate, or full member. But because most people around the world don't understand what that is, we have a relationship with Cornell. And so I also have a professorship at Cornell, which is the medical college across the street from Sloan Kettering. Now, here are a few more things that have come in mind during my time back to Sloan Kettering. And this is one of my favorites. And I hate when people say we've always done it this way. If you go in the operating room and somebody says, well, I've done this way for 20 years, I usually say, well, it may be about time to start thinking about changing. If you stay doing the same thing for 20, 30 years, that's not progress. That's just being stagnant. So I always tell folks to never stop asking why. Be very critical of expert opinions. I am now one of these experts. I sit on these guideline committees. The these guideline committees sometimes put out guidelines, which I absolutely do not agree with. So I think you need to always be critical and always ask why, even if things have been done for 30 years. And at least in surgery, there's a couple of examples of bowel preps, not letting people lead until they pass flatus, all these other things that we did that were just really made up standards, if you will. And so I question everything, even the pap smear every year, that people still do, that's still silly. So I think that you need to always question. And Dr. Murray Brennan, who's a chair of surgery here, said something very important. He goes, you don't have to do these fancy research and it has to be this high level. If you can answer one question that's clinically relevant in a randomized trial, you've succeeded. It doesn't have to be something super fancy. So I always go with ask questions that matter to help patient care and will have impact on patient care, not just to fill your CV up. Make sure you care about the question. If you don't care about what you're asking, don't do it. You're wasting time. And many of the projects I've worked on come from my daily clinical and surgical events. As I see patients, I'm like, well, is this making sense? What can I do better? So I'm just going to go over some of the areas I've done research in and show you some examples, but I can't cover all of it. So one of my biggest things as a surgeon, especially in running the MARS program, is and just perioperative. I have a big interest in improving perioperative care, which has nothing to do specific with GI oncology, but it's one of the areas I like to focus on. It's the critical assessment of devices and products, and it's actually part of my role as a director for MIS. And here's an example of one of my first publications looking at a device, which was the separate film. And, we were using this a lot. And then, again, another example of something stemmed from a clinical question or concern. We were using this a lot to prevent adhesions. Everyone knows after laparotomy, a lot of scar tissue forms and adhesions can then be a lot of problems. It leads to bowel obstructions. It can lead to very difficult repeat surgeries. And these are patients with cancer, so they usually need repeated surgeries. So we started using this a lot. But then some of the partners in the group said, I think I think it's causing more post-operative collections. I'm like, no, it doesn't. So, it's an example of you can't just go based on what you think is happening. You really need to sit down and analyze it properly. So we did. This was published. This is looking at it. And what we found is that there were more collections actually in those that were using the separate film product. This is 423 laparotomies that we looked at for ovarian cancer debulkings back in the time. We published this in 2009. And what we found is that the rate of any abdominal collection was higher, or nearly fourfold higher, in those that we used the separate film product. And the infected collections especially were very high compared to that where we didn't use it. So, and it required more interventions, more procedures afterwards. So because of this study, we no longer have used this ever since that time for cytoreduction. Again, I think an example of a question or a concern was brought up clinically, anecdotally. We didn't just say, oh, forget it. We actually sat down and looked at it. We found that it was true. And we made a decision and changed our practice. Not a groundbreaking study, of course. I mean, it's not that some cancer cure, but it still just highlights the importance of asking questions that matter. If they do, this study was done quickly. And another question that came up is, people were always in the operating room trying to figure ways to reduce wound complications. These things on the left here, when the wound breakdown is very painful. This is a patient who, because of all the multiple wound changes, developed a severe rash all around. I mean, this is very uncomfortable. So there are a lot of products and companies trying to come to us. And there was this Preveno, they do the wound vac system. They created this prophylactic negative pressure wound system. And, the company saying how great it was. And there were some studies that it reduced the wound complication rate, but there were no really good randomized trials specific to GYN laparotomy. So we said, let's see if it works. And here we said, we're not just going to use it and look back. We're going to do a randomized trial. So this is a randomized trial that I did and was published recently in the Green Journal, Obstetrics and Gynecology. So we actually did a randomized trial, got some funding from the company. We randomized nearly 600 women undergoing laparotomy for gynecologic malignancy. So just at our place and 3 other centers that work closely with us, but the majority were our institution. So this is also, again, full circle from my very first randomized trial as a resident that was complete disaster to one here where I did now with all these years of experience in research and sort of understanding the design, a trial that randomized almost 600 women, completed, done, and found that the rate of SSI wound complications was exactly the same whether you use the device or just standard gauze. So because of this, we've also stopped using this product. So I think that's, really, if you're going to do useful research, that's how you need to do it. And you can, in the molecular world, you can do that also. You can ask those questions that help us take care of patients, that hopefully find better treatments, maybe help us select for better things. So another area of devices on a bigger scale is the robotic system. I've been doing laparoscopy. I was trained in laparoscopy. I think minimally invasive surgery is the way to operate. It's better for patients. And this whole robot thing came out during my fellowship. We had one of the originals, but we never used it. I learned it in 2006. I've used all the platforms. And the way I view the technology is that it's a natural evolution of MIS. So we go from looking down the scope to looking at video monitors to sitting at a console to operate. None of this is anti-laparoscopy. So you also have to have a perspective when you ask your questions. I think there's been a bad perspective of assessing the robot by comparing it to just laparoscopy. So what we started doing is this as institutions support us. They want us to build a program, but we kept a database of every single case. And we still have that database. It's got over 8,000 cases now. So the first thing we did is, after we had a few cases done, we actually looked at what happened at our institution once we introduced the robot. We noted that there needs to be a learning curve. We also realized that the complication rate is actually no worse with robot. There was a lot of stories back then saying the robot had more complications than it actually doesn't. It's slightly lower. And for grade 1 to 2, it's even lower than laparoscopic cases. And what we also noticed here is that we were sending patients home the same day now. Started to say, why are patients staying home, staying in the hospital? This led to a publication a few years later saying that we can send patients home the same day. And we looked at it. Our first 157 same-day discharge, they were doing just as well as those who would go staying overnight after minimally invasive surgery. And so now our standard is to send people home the same day, even after a robotic radical hysterectomy. We looked at then, how is this robot impacting our care? And in morbidly obese patients, what we noticed is that by introducing the robotic platform, we decreased the laparotomy open rate significantly. It went from 90% down to 20%, simply by the introduction of the robotic platform. What we also realized is that because of this, we're saving money in this cohort of patients, because the rate of complications went from 36% to 15% in all patients who are morbidly obese. Now, you don't compare just laparoscopic cases. You take the whole group of morbidly obese patients who come to you, you introduce a device, and then you see what happens to the whole group afterwards. So since you do more minimally invasive surgery, you have less complications, you save money. You don't compare the small number of laparoscopic obese patients you did before the robot to now the 80% of robotic obese cases, because the ones you did before are very easy. You look at the whole cohort. That's how you look at it. And then we also looked at cost effectiveness for all uterine cancers, and we found that it was a cost benefit because we increased our MIS rates in endometrial cancer from 50% overall to nearly 90%. It's almost 100% now because of the robotic platform. So that's how you assess the device properly. And this is because of our research. This is our growth of the robotic platform at our institution. We now have 9 robots, and we're about to get 2 more. And now we do about almost 1,000 cases a year, just GYN cases. And this is all because we've done it safely. We analyze it, we report it, and the institution keeps supporting us. In addition to this, I've had many other interests, all clinically derived. We've been a big proponent and actually the leaders for sentinel node mapping for uterine and cervix cancers. Our work is the reason the NCCN guidelines in the US have changed and incorporate our algorithm. I've been fortunate to be a part of this research and have multiple publications looking at the use of this sentinel node mapping. And the very first one was how you start at the beginning. So when we first started introducing sentinel node mapping, how does this impact our detection of nodal disease in stage 3C cases? And we found that it did not. Taking out less nodes, more directed nodes, we still have the same rate of stage 3C disease. We then start to see, can we find them better? It used to be blue dye and technetium or blue dye alone. We then start to introduce indocyanine green, and now that's become our standard. Then we have to have other questions come. So we find sentinel nodes. Now we have low volume nodal metastasis that we didn't deal with before. How does that impact outcome? Then we started to work with others. Another key principle here is you need to collaborate often, especially if you don't have all the patient data. We do a lot of research with a lot of colleagues from both in the US as well as international. And I have papers with international colleagues. And these are some of the big papers we did with the Mayo Clinic. We compared our sentinel node mapping patients to their lymphadenectomy patients. And we had multiple publications basically saying that sentinel node mapping is sufficient. The role of lymphadenectomy, we looked at that, and those had nodal metastasis. And again, continued work with our Mayo. These are all papers we just recently had published with the Mayo Clinic. And if you notice now, you start to see my name less and less at the beginning. Actually, you don't see it at all in the beginning. Here, that paper it is. And then all these other ones, you see my name mostly at the end. So I've been, for many years now, I rarely do a first author publication. And this is all guiding my fellows, and every fellow who works with me and does the research, they get to present the abstract. They are up there and they get their name as first author on all these papers, and they have all gone on to be very successful academicians. And then just my last sort of thing to talk about research is this whole thing about minimally invasive for cervix cancer. It's not a talk on that, but again, an important clinical question that was prompted by a randomized trial saying that minimally invasive surgery is bad. The first thing we did, well, we don't want to hurt people. Let's stop. Look at what's happened here. At an MSKCC, for the LAC trial eligible patients, we had zero difference in outcomes if they were done MIS versus open. The MIS curves are the blue line. So actually in overall survival, it's a little higher here. So we stop. We don't want to hurt people, right? Never want to hurt people. But if we see some publication that says otherwise, we need to stop right away. People need to look at their own outcomes also. Not only do we do that, we then did a multi-center study. Again, the importance of collaborating with 15 centers. These are all cases that were eligible for the LAC trial. You can see here there's 1,000 cases. Propensity score matching, another thing I've had to learn along the way, which is plus or minus on that. It's not so great either. It's no different than multivariate logistic regression. But in this multi-center of 1,000 cases, there was also, when we matched them, no difference in MIS versus open. Now, this is retrospective, of course, but just ask the literature. And all this has led to the next randomized trial that I'm involved in, which is a randomized trial comparing robotic versus open, which we have addressed all the LAC trial issues that we think. I'm also mentoring a junior faculty from Ohio State, so from another institution even. I think that's important. On the co-PI, we're working together, and this trial should be open. We got about $9 million from Intuitive to do this through the GOG Partners, which is our cooperative group. And this trial should be open by end of October and November. So who can do it? I think everyone can do it. I think if you stick to the basic principles of loving what you do, have meaning to your research. Don't just do it because the hardest thing is if you have someone who's a senior telling you to do this research project and you really don't want to do it, it's going to be unsuccessful. You'll be unhappy. I tell all my fellows, if any faculty ever asks you to do a project with them and you really do not feel like it's appropriate for you, you need to speak up, or I will speak up on your behalf, because otherwise it will be unsuccessful and not be done correctly. Always keep asking questions. You ask one question, you answer it, more questions come out of that. So keep it going. Now, how do you do this in a busy schedule? I mean, like today's one of my clinic days, so I'll be seeing patients shortly. So, you have to sort of set aside time on a schedule. For me, I have an academic day, which is Thursdays, tumor board in the morning, and then I have the rest of the day to do academics. But with my administrative roles as MIS director and fellowship director, that eats into it. But I try to put blocks on my schedule, say research or work on this. I actually list work on this project. I schedule meetings with the fellows and their research. And then, you really, with a busy schedule, I operate 2 days a week. I have clinic 2 days a week. I do things like this in the morning. I get in at 7 in the morning, 6:30 in the morning, and I work on things. I try to work on things in between cases if I can, especially in the operating room. So I'm always trying to do things. And I actually every day will have a little post-it that says, these are the 2 things I need to get to today in terms of my research. I would say you need to collaborate. Obviously, it's become easier for me to publish because of the many junior faculty and trainees that I'm now mentoring as their senior author. One thing I don't ever want anyone to do is waste weekends and time with family, friends. This is a very difficult life we have as a busy G1 oncologist and busy surgical oncology practices. It's difficult, it's challenging. You need to have downtime. So I never do anything on the weekends. And I always, if my daughter has a volleyball game, my son has a lacrosse game, I make it so I get to those things. And that will keep you happy and successful. So again, I know it's very hard, but it can be done. And if you're doing things you love, like I said, it will work out. Your work is going to fill a large part of your life. And the only way to be truly satisfied is to do what you believe is great work. The only way to do great work is to love what you do. If you haven't found it yet, keep looking and never settle. So thank you for having me. I hope this is what you were hoping for and that this was somewhat helpful.
Mario,
yeah.
Fantastic. Thank you so much. You told us very, very important issues about to join a clinic and academic career. Fantastic. And I'd like to congratulate you, and I'd like to see if someone has a question or comments. Fantastic. I think that you told us very important issues. The first thing that I thought is keep asking why every day in your day, during your activities, professional, why. And I can tell for everybody that Mario Leitão is one of the leaders in my area, gynecological oncology. And the most important papers in my area came from Memorial, from gynecological oncology department. And Mario, I could see in my career that the Department of Gynecological Oncology and Memorial, you publish a lot of retrospective studies with big samples and very organized. And for me, it's fantastic because I like to know from you how in your department you organize your databases for publishing a lot of retrospective studies, because for me the memorial is an example in this.
Yeah, I have the same question. Sorry. Before you answer, Mario, thank you for the lecture. How do you save time for doing that? And what have you changed in the past 20 years in your daily practice just to organize and structure your database? Thank you.
Yeah, so we've been very fortunate here at Memorial since the 1940s when Dr. Brunswick was here. He had Dr. Virginia K. Pierce was also here with him. They realized early on the importance of properly maintained databases so that you can know what you're doing to patients. So since the 1940s, we've had an ongoing collection of patients in we can't call it prospective, but we have databases that patients are entered on a regular basis for every disease site. It was started by Virginia K. Pierce, and back then it was on paper. So when I was a fellow, we used to get go into the Virginia K. Pierce office, which she passed away, and there's boxes and boxes of patient information for every disease site. That's obviously over the years morphed into electronics. And so now, what we do for every disease site is we have and this costs money, we have to pay people, but we have research staff assistants who screen all of our clinics for every patient that comes in with every complaint. They then follow up to see what the final diagnosis was. And we maintain a database of every single patient that comes into Memorial Sloan Kettering in a research database using REDCap. So that now is a database that we've built on before. So this started more of this prospective where we pay people, research staff to do this. It started about 2015, 14 for all of our disease. We have one for ovary cancers, endometrial, cervix. The vulva and vaginal kind of get thrown in with the cervix. So for our three main diseases, so every single patient's captured. We put some minimal information on there, but what happens is then as we do questions on that, we ask if there's more, if a fellow goes back and looks up other things that weren't in that database, that gets backfilled into the REDCap database so that the next person that comes, the information is all there. You just have to ask your question. So that's something that requires a lot of coordination. It requires some funding and some dedication. We have research fellows also. So, I mean, if you want to use, not pay an actual research assistant and the salary and all that, if you have fellows that are doing research, we assign them to one of the databases. It's their job to identify every patient that comes. And it's not just in our GYN oncology clinic. They screen the radiation oncology clinic, because there's only a few people that do GYN, and all the GYN medical oncology clinics for all these patients. So we identify every single patient that comes to us with uterine cancer, every single patient that comes with cervical cancer. They're all put onto our database that now has thousands and thousands of patients.
Fantastic.
And this is also a principle that Dr. Murray Brennan in surgery started. he had a dedicated team for sarcoma, a sarcoma database, which became one of the largest sarcoma databases in the world. And they've published extensively after that. So many of us at MSKCC across all services have modeled that, where we have active entry of patients onto database, which some people will call registries. That's a typical example of a registry where you enter patients onto a research database. So that's how we do it.
Fantastic, Mike. I'd like to see if we have questions from the students or professors, please, for Mario Leitão.
Ricardo, we have a question in auditorium. Ricardo?
Yes?
We have a question in auditorium.
Okay, please.
Hi, good morning, Mario. It's Carlos Andrade. How are you?
Good, how are you?
Fine. Two questions. First, about your research staff. What's the size of your own? You have your own research staff for you?
Yeah.
Or is for gynecological oncology staff? Yeah. Or is for all Memorial Sloan Kettering that you compete?
Yeah.
Or is it staff?
So,
and second, how you can collaborate with your research? What's the impediment for us to collaborate in your research?
So the first question, our research staff, we have our own dedicated GYN surgery research staff. So we have a manager, someone below her, and I believe we have about 8 research staff assistants, 8 or 9, just for GYN surgery. I don't have my own. None of us has our own. I have 12. I have 11 partners. There's 12 of us. So it's for all the division of surgery. Now, each research staff assistant is assigned. One is assigned to the ovary database. One is assigned to the endometrial database. One's assigned to cervix. And then we also participate in the GOG NRG trial. So the work is decided by the research manager, who's just for GYN. And then she reports to an overall research director. And so it branches out from the overall department of surgery, and every service has their own research staff group. No one's specific to me. In terms of the fellow research, usually they get involved in providing like the database, access to the database for retrospective things. They make sure that also we get our IRB approvals in. They maintain our waivers for retrospective research. So we have a dedicated staff of about 8 to 10 research assistants for all of GYN. There really is no impediment to working with us in terms of retrospective. I mean, it depends on what kind of research you're looking at, retrospective versus prospective. We've done research with folks around the world. The bigger impediment is not so much us, but it's usually the getting the data to us from other centers. There's data use agreements that have to be signed, go through our IRB and the IRB at the institution. What we realized recently, we did some work with the folks from Norway and Italy, and it was just them getting their IRB to approve the data use agreement to get us their data. So that's really the only impediment, is the legal process of the IRB based agreements, where obviously you can't share certain things, at the US because of HIPAA. You have to be careful with kind of patient identifying information. So other than that, other than the logistics of the agreements that need to go into place, once that's done, it's pretty easy to do research with us, and we're open to anything if it makes sense to us. I mean, if it's something that we haven't done already and it makes sense to us, we're always open to working with anybody around the world. And then if it's a prospective study, it's a little more challenging because, there could be regulatory things. The consents need to be in the language of a different, that makes sure we have an approved consent. So there's other issues that come up with prospective based studies. If there's funding, getting money, if there's tissue that's needed for sort of translational work, that's a whole other area that creates a lot of more logistical problems. And we've tried to do that with Brazil in the past. And back a bunch of years ago with whatever sites we work with, it was very challenging to try to get the Brazilian government to agree to send us tissue for some of these prospective cooperative group studies for translational work.
Fantastic.
Congratulations. It's the first time that I see your work, and It's been really amazing, your vision, your track, so congratulations. And I think everything you said was really important to us. We are a very busy hospital, but always striving to do research. And it's not easy to find time between, a very busy hospital with thousands of patients arriving every day. So, but one of the things for me that you said is really important is that we have to choose questions that are important to our daily life, right? And don't need to be very fancy. I work with genetics, but I realize that sometimes very simple questions can have very important impact. And don't need to use the next generation of equipments or genetics to fulfill that. And you show us wonderful examples of simple things, but really important that they translate in the daily life.
Yeah.
Another thing that I like to have your opinion is that frequently, when we don't have time, we try to, you others questions what the others want to do with our cases. And I think that is the wrong way to go, because nobody will take care of our problems. If you ask someone even from Sloan Kettering, so can help us to solve our problems, you have to know what is our problems.
Right.
And then the research, so we have to work. So what I'm trying to say, we cannot delegate to others what is the important questions that we found at our place.
Yeah.
And of course, collaborations are key for all of that. And Sloan Kettering is giant, and even so, you showed us that you work with a lot of partners. But I really liked your point that it's really fundamental to work with the problems that you have at your place, and then try to find collaborations way to solve that. I have one question. So you have one day for an academic. Every staff that you have is one day is granted this academic day, or they have to deserve it? Because we are trying to discuss with the direction of our hospital. Yeah. It's always a balance. You have to attend more patients, but also have to do research. So how is this done at Sloan Kettering?
So you have to have, the hospital has to have a mission that they want to do research. If they don't, then it's going to be very difficult. MSKCC's one of its main missions is to do research. So pretty much every surgeon, and we're talking about surgery, medical guys have a lot more time, actually, to be honest with you, but us as surgeons, because we're also expected to operate and bring in the surgery, but we don't ask for it. We are all given at least one academic day. Now, we have folks who are basically also surgeon scientists in the lab, and they have their own labs, or they work in a lab. Those folks actually will get 3 academic days and only see patients one day and operate one day. So those are some of our surgeons in the Department of Surgery are actually working in a lab. And so those folks only have clinical duties on 2 days, and then they have 3 days to work, to do their research in the lab and such. But that's a true surgeon scientist in the sense of doing molecular oncology research, which I think is a challenge. That's something I could have done if I wanted to, but I just love operating too much and only operate one day a week. So for the rest of us who are clinical researchers, if you will, everyone is given an academic day, because the expectation from Sloan Kettering is that we are being academic productive, and our evaluations every year by our chiefs, one of the big parts is how much research have you done? What have you done? Are you a thought leader? We get points for that. So, and that points then relates to our compensation, what I get paid. So, are you a director of this? Are you a director of that? So you have to have, the hospital has to have that as a mission, that the hospital wants to do quality oncologic research. If that's what they want to do, then you have to let them know, I need an academic day every week so I can settle down, regroup, and work on my research and work with my fellows, my trainees, my postdocs Otherwise, it's nearly impossible if you don't have an academic day. And again, if you do questions that you love, like I said, you want to, in between cases, you want to go back and finish it, because you're interested in the question, right? You want answers to see what the answer is, right? Some of the projects that take me the longest is when someone else asks me to help them. I'm like, I'm not that interested. It's just you procrastinate on that. But if it's something I'm very interested in, I'm always going, let me look up some more. Let me see what the fellow did. Let me look at their data. So, but yeah, the hospital has to have a mission that the research is important to the institution. And then in order to do that, you guys have to let them know that we need, at least busy clinicians, we need at least a day or half a day, even if you can get that, to just work on my academic stuff.
Perfect, Mario. If someone has a question, Rick and other professors, students, please. We have, it's 9 o'clock, we are finishing. Okay, Mario, I think that we do not have more questions. I have to say thank you so much, my friend. I will contact you for rock trial. We are interested here in the interview because we have robot here, and if he give us the opportunity to operate robot surgery every week here. Fantastic for public patients. And I'd like to say thank you so much. And we learned a lot, mainly with the main issue that you told us, why? One question. From one question, we develop a paper. Why not? We told us, we show us very well, and focus on methodology. I think that it's another message that you told us. Focus on message and learn statistics. I love it. And thank you so much, Mario. Thank you so much.
You're welcome.
I have no words to say. Thank you, my friend. Thank you so much. Have a good day.
Pleasure.
Bye.
You too. Bye, everybody. Thank you.
Thank you, Dr. Mario.
You're welcome. My pleasure.
Perspectiva.